talk on adapting concepts from cellular automata to explore bushiness and convergence in Boolean network dynamics. So he comes from IMC.
First, I would like to thank Professor Arijit for giving me this opportunity to present this work. So the title of my talk is Adapting Concepts from Cellular Automata to Explore Bushiness and Convergence in Boolean Network Dynamics. So at first, I will just go brief like what a Boolean network modeling is. So it's a discrete dynamical modeling framework that has been used extensively to model gene regulatory networks, where the nodes of the networks corresponds to the genes and the edges corresponds to the regulatory interactions among the genes. As Professor Sitabro mentioned that the gene expression we can actually approximate in terms of on or off, that is 1 or 0. So here we can mention like that. And we can assign a function, a Boolean function at each node that govern the dynamics of this network state. So like for any n node network, we have 2 to the power n possible states, and those function determines that what is going to be the next network state for a given initial state. And these state transitions can be mapped into a graph that is called the state transition graph. So starting from any network state, one can reach to a state, to a fixed state that upon applying the functions again, it doesn't change the state. Those are called the fixed points, and it can also fall into a cycle as well. So one such combination of functions is what we'll refer to as a model, and a network can have like a numerous number of models, and also we can have numerous number of models that can lead to specific fixed points. So these fixed points are of biological relevance because they are actually associated with the steady cell types, the gene expression of steady cell types. So now let us see by an example. It is a gene regulatory network of myeloid differentiation GRN. So let us consider 2 models for this network. So we are not changing the network structure, and the interactions and the genes are the same. We are only changing the regulatory functions that we are assigning at each node, and that I have mentioned as integer form of that Boolean functions, and they both give the same biological attractors as we expect for this specific myeloid differentiation model. So what is the state transition graph for these 2 models? If you see the state transition graph for this first model, we see a bush-like structure. So it is very bushy in nature. But for the STG of model B, clearly there is a visible difference between these 2 state transition graphs. So the question is, can we quantify this difference? Though they are giving the same biological attractors, but their dynamics is completely different. So how do we distinguish this difference? So we searched in the literature of Boolean networks. We didn't find a measure that explicitly distinguishes between these structural features of these 2 networks. So then we looked into other possible discrete modeling framework. So we searched literature, and we found that in cellular automata theory, specifically the study by Andrew Wunsch, he has explored these structural attributes very extensively. So in cellular automata, so this is a specific type of cellular automata. It's a ring-like structure, 1D structure, where here each cell can take the value 0 or 1, and they take inputs from its k neighborhoods. And at each cell, there is a homogeneous logic rule that actually governs the dynamics of that system. And there he mentioned that a highly bushy state transition graph that has a shorter transient show order dynamics in compared to a less bushy STG where we can see an chaotic dynamics. So here they have here he has quantified the measures by which we can actually structurally differentiate between different types of state transition graph. So we inspired from these ideas and try to explain this in case of a Boolean network. So it is an example of a 5-node, it's an example of a STG of a 5-node Boolean network. So you can see that there are nodes, those are colored in orange. They are called the Garden of Eden states. That is, they are the nodes, and that don't have any integrity. And there are certain nodes. The other nodes are called the non-Garden of Eden states. So the first quantity is based on the Garden of Eden state. So the fraction of Garden of Eden state in the state transition graph is called G density. A more intuitive measure would be in degree. So for in degree, we take the average in degree over all the non-Garden of Eden states. That is what we call as the average in degree of the non-GOE states. And that is, this is the formula for that. And there is an explicit relationship between this in degree and the G density that can be written in this form. So any one of these two measures can be used as bushiness measure. The one other quantity that we are interested in, that is the transient time. So that is the time it takes to reach from any state to the fixed points. So here if we consider the state 13, this GOE state, it takes 4 time steps to reach actually the fixed points. So in this way we can compute the transient times for each of the Garden of Eden states, and we can take average of that, that is the average transient length. If we take the reciprocal of that quantity, that we say as average convergence rate. So now we have the quantities that we can associate with some state transition graph. So let us see where do these two state transition graphs stand in terms of these two quantity. So you can clearly see that for all these 3 quantities, the model A shows higher value in compared to model B. So here I would like to mention that the function that we have chosen for the model A and model B were not all random, but for model A, the functions were from a specific class of function, that is nested canalizing functions. And that is known to be a biologically meaningful functions. I will tell in the next slide. So the main question we ask that do biologically meaningful functions lead to a more bushy and convergent STG? Because in recent reconstructed Boolean modeling network dataset, it has been shown that some classes of functions are more preponderant that it actually should be theoretically. So there are different types of Boolean functions like effective functions, effective and unit functions, read-once function, nested canalizing functions. I'm not explaining the definitions of all of these, but I will just mention that these 3 types of functions are known to be biologically meaningful functions, where the effective functions actually almost capture the whole state space of a Boolean functions and can be considered as a random Boolean functions. So now we have the quantities that distinguish the structural features. Now we know that these are the biologically meaningful functions. Now we want to check whether these biologically meaningful functions leads to more bushiness or not. So for this now we need to generate ensemble and functions. So we have the network structure given. We know that these are the expression states for some specific cell types. So we constrain the truth table of the function using these biological fixed point constraint, and then we constrain the truth table with the required type of Boolean functions, and finally we generate the list of allowed Boolean functions at each node. And in this, following this flow chart, we actually generated 4 ensembles of models: EF, UF, RF, and NCF. And we generate 1 million models for each of this class, where we have fixed the network structure, and all the models in any ensembles gives all the biological attractors. Now for our analysis, we actually considered 10 gene regulatory networks from published literature, where I am going to show the results only for the first network, that the same results holds for all of the other networks as well. And so if we see the distribution of the G density and average convergence rate, we see what we actually expected. We see that for UF, ROF, and NCF, the value of G density is higher in compared to effective functions. Even inside the biologically meaningful classes, we see that there is a slight increased value in case of the NCF ensembles. And for the convergence rate as well, we see a similar trend. But computing these global measures become computationally very expensive when you go for larger networks. So we asked, is it possible to obtain more feasible proxies for that? Then again we looked into the literature of the cellular automata, and we found that Andrew Winshire has actually proposed a parameter called Z parameter. So it is a local descriptor and it correlates well with the G density. So it is computed from the rule table directly. One can compute the Z left and Z right while considering the inputs from left to right orientation or right to left orientation. And then one can take the maximum of these two values, and that is what the Z parameter for some specific rule. But how do we adapt this in case of Boolean networks? So we see some challenges. If we see the similarity and dissimilarity between the features that actually distinguish these two dynamics, so for both of them we have considered the synchronous dynamics, and the time step is also discrete. That is what allowed us to directly adapt the measure, the global measure, that is like the G density or in degree, without any constant. But when we want to adapt something in terms of the rule levels, then we see that in case of input, in case of CA, it takes input from its k nearest cells. But and for all of the cells, the number of inputs is constant. But in case of Boolean network, the number of input is not constant. And also if you see that in CA, at each cell, the regulatory logic rule is homogeneous, but that is not true in case of Boolean network. So how do we deal with this problem? So in CA, we have seen that we can take two possible orientation, that is from x1, x2, x3, x3, x2, x1. So in case of Boolean network, we can take all 3 factorial permutation if we take a 3 input network. So here we have two quantities. We can take the maximum of those, that gives the Z parameter. Here we explore other possibilities as well, the maximum, minimum, average, and the average of the maximum and minimum. To go to the network level, we take the average over all these functions. The other quantity is network sensitivity. I am not explaining it in detail. So here as well, like for a function, we can define the network sensitivity. At the network level, we can take the average over all the network sensitivities. And finally, if you see the correlation, then we can see that the Z parameter here also like correlates well with G density. Specifically, if you see all 10 models, across all 10 models, the last 3 local descriptors, that is the Z average, Z mid, and the network sensitivity, show good correlation with G density, but it is not so with the convergence rate. So for convergence rate, we actually provide an estimator just by taking some random state and not considering all the Garden of Eden states. So in summary, we found that the biologically meaningful functions lead to higher Bushy and convergent state transition graph. We adapt the Z parameter that was defined in cellular automata to a more generalized dynamical system, that is Boolean networks. And the average sensitivity and some versions of the Z parameter can be used as good proxies of bushiness. And the average convergence rate, for average convergence rate, we gave a random sampled base measure that can actually. So I would like to thank my PhD supervisor, Professor Arijit Samal, and my collaborators, Professor Olivier Martin, Ajay Subarayan, and Soumitra Kulkarni. So this talk is based on this recent preprint. And for a more detailed discussion, please visit to my poster. And thank you.
Right. Thanks a lot, Priyotosh.